This lecture is an introduction to asexual propagation, different than propagating through seeds, which is a sexual means of propagation. This is done using parts of the plant that are not the seeds. In asexual propagation, we take parts of the plants like stems, sometimes leaves, roots, and we cut them off of a parent plant, and we induce them through different methods to grow roots and leaves. So the different types of asexual propagation involve cuttings. That's the most common, taking cuttings of the stems, sometimes roots, sometimes leaves, but mostly stems. Take a cutting of a youngish to semi-young branch, cut it off, put some rooting hormone on it, put it in some media, some potting-like soil, and give it some bottom heat, and after a few weeks little roots will form off of what was once a stem tissue of a plant. So obviously roots don't grow off of the branches very often on our trees here and shrubs, but you can induce that stem tissue, those branches, to grow roots in different ways. Layering is another method. Divisions, budding, grafting, and tissue culture are all the different methods of asexual propagation that this lecture will explain in a little more detail. And then after this lecture, future lectures will be devoted completely to each of these topics, just about. So this is just an overview before we get into the nitty-gritty of spending a lot of time on cuttings and layering and etc. So let's start off with talking about cuttings. There are three main types. You can take cuttings from stem tissue. That's the above ground branches and stems of plants. And that's the most common type of cutting that's taken. But you can also take cuttings off of leaves, just leaves, and induce those to form roots and new leaves and shoots. You can also take cuttings of roots to grow and propagate new individuals of that same plant species. With cuttings, there are, it depends what you're trying to propagate. For the type of cutting you'll take. Cuttings can be taken from really new tissue on a stem. Those are called softwood cuttings, like the new active green growth on stems, like the very tips of stems of woody plants. Those that are younger tissue on a shrub or a tree that you're taking cutting from, the younger tissue has a higher potential to form roots than older tissue, and but it also has the lowest survival rate because it's young, it's not hardened up, it's still kind of vulnerable and green and juicy, and it dries up easily and rots easily. So the younger the stem cutting, the higher potential it will form roots because it's in growth mode, but it's also the ones that don't usually survive as well. So you have to kind of balance out between survival and root growth. Some plants, one gets more success from taking greenwood cuttings. That's the tissue that's not the very tip of the stem, not the very newest active growth, but a little back a ways on the stem. Slightly firmer, a little brownish. You can still get good cuttings from that and propagate lots of plants through greenwood cuttings. Then there's semi-ripe. So as you see from the top to the bottom of this list, the tissue's getting older. We're starting at the tip of a stem and we're going back towards the next main branch, so it's going into older and older tissue. Semi-ripe cuttings, they're even firmer. They're usually pretty much brownish, reddish, because the new growth is green, and usually they brown up as they get older. The firmer stems with buds that have begun to develop can be good for those type of cuttings, and heel cuttings with more cambium can be taken too from semi-ripe cuttings. We'll talk more about heel cuttings a little later in another lecture. Hardwood cuttings, that's some plants, if you take softwood cuttings from them, like for example, I'm going to be taking some cuttings from some sycamore trees near my home in some wild sycamore trees because I want to plant some in around my home. And to propagate these, if I took softwood cuttings, during the spring, the new early growth, none of them would survive and they wouldn't form roots. The sycamores just can't be propagated with softwood or greenwood cuttings. A lot of our native tree species, especially the ones that are deciduous, can be propagated with hardwood cuttings. So it's older, what you would call dormant wood or ripe wood. It's brown, hard, and it takes a long time for it to root, but they do not dry out and they don't rot. So again, it depends on the plant you're trying to propagate, of which of these maturity levels of cuttings you would choose to use. And we'll learn all that as we do quite a few different species in our class. Some other tips, methods used in using stem cuttings: when you put them in some kind of media, it's a type of like soil, it should be at least 4 inches deep. It depends on the type of cutting you're taking and the size of it, but it has to be deep enough so that you can insert that stem into the soil and it can hold up the actual stem, and you get a couple of growth points below ground, and those are called nodes, which I'll talk about in a minute. The best time of day to take cuttings is early in the morning because plants have more moisture in them. So if you can, take them then. There are plenty of plants where a lot of these little details don't matter that much. Some plants you can take cuttings from almost any part of the tissue of the plant, no matter what maturity level it is: green, young, old, brown. You can take them in the morning, in the afternoon, at night. And they're so hardy and they root so readily that it doesn't matter that much. As you do more propagation, you'll find out some species are more challenging and require more detailed methods to make successful cuttings grow from the mother plant. When you take a stem cutting, taking a branch off of a plant, and the one of the cut ends, the lower cut end, which is closer to the ground, or the main stem on the parent plant, you cut that and cut it off of the parent plant, and often we'll dip it in some kind of rooting hormone on that part that's put into the potting media to help induce it and speed up root development on that cut stem surface. Sometimes those rooting hormones also have a fungicide in them to prevent rotting. These are just 2 examples. One is Rootone, another is Dip and Grow. They both have ways of inducing root growth. A little overview of hormones. The hormone that is used in those rooting hormones that are put on the stem cuttings, those are a variety of hormones, but mostly auxins. That IBA, indole butyric acid, is a rooting hormone that's used. Auxins, you're seeing it here second on that list, is another type of plant growth hormone produced by a plant that promotes stem elongation and root growth. So the IBA that is in that rootone is an auxin type of hormone, and it will increase development of roots off of the cut stems. There's a list here of other different hormones that plants produce, and for various reasons. One of the other well-known ones down kind of near the bottom is called ethylene. That's something that fruits produce on their own to help ripen themselves, and that's why if you leave your avocados or whatever you're trying to ripen, bananas, something like that, and get them to ripen quicker, you put them in a bag, and the bag fills with ethylene gas, which is produced by the fruit itself, and that further ripens that fruit. Little side note here, if you look at the very bottom, auxins, those hormones that promote stem elongation, root growth, fruit growth, those are produced in the leaves and the canopy, and when they stimulate root growth, they have to travel all the way down. You can see that little red arrow at the bottom diagram. So the auxins are produced in the leaves, and they have to travel all the way down to the root tips to stimulate the root growth. Another class of hormones are the gibberellins, and those are produced in the root tips, and they stimulate canopy or leaf growth. So it's almost like the hormones that are produced in a plant are produced far away from where they're actually taking action. back to our types of cuttings: stem, leaf, and root. The second one is leaf cuttings, and that's just using sections and pieces of leaves to reproduce new plants. Some plants you can do this with, not all, but sometimes things like on begonias, seeing that in the image there, or African violets, if you cut a leaf off of a mother plant and you turn it over and you make little cuts or nicks through the main veins on the underside of the leaf, and then put that leaf flat on back onto some rooting media, roots will grow out of those cuts on the leaf. And if you have a bunch of them, little areas where roots are growing out, those can all be cut apart and made into individual begonia plants. So it's a way to propagate more just from leaves and inducing roots to grow off of the leaf veins. Root cuttings can also be taken to propagate plants. The use of roots, the roots are dug up, cut up into small sections, depends on the species, how big of a section you need. And those are then put into their own pot and media and induced sometimes just through water, sometimes with bottom heat, sometimes with a little rooting hormone to produce their own smaller new roots and new plants. Bamboo is a good example of a plant that is propagated using root cuttings, usually taken in the winter, and that's pretty much the only way you can do it. Sometimes you can do it by divisions, pulling the big clump apart, but if you want completely new plants and small plants, you use root cuttings. Layering is another technique of propagation of plants by asexual means. So far we've talked about cuttings, and our next method is layering, and that is taking plant stems or branches while they're still attached to the parent plant and inducing them to form roots. So it's a natural process for lots of plants. Plants who have branches that get so long that they kind of hang back down onto the ground, and where they touch and stay in contact with the soil, new roots form. You've seen this probably in things like berry bushes like blackberry and raspberry, these long vines, and whenever they kind of touch the soil, they form roots and a whole new little plant forms there. It's a way of these big bushes can sometimes spread. But it's a natural process, but we can take advantage of that natural tendency and force stems to produce roots by burying those stems while they're still attached to the mother plant, burying part of their stems and wounding that buried part first, and new little plants will grow off of those areas. 3 types of layering: air layering, trench layering, and mound layering. Air layering, sometimes called Chinese propagation, is where an above ground stem of a plant is taken. It's left there on the tree. You just find a good spot, cut the bark off all the way around that stem, and then pack some moist media, sometimes sphagnum moss, peat moss, and then seal it with a little bag. So you're enclosing that wound in a moist environment, and the plastic put around and tied off is so that the moisture doesn't seep out, and it keeps pathogens out too. Over the next couple of months, that thing that was a branch suddenly has moisture on it long enough that it's been wounded, so those tissues right underneath the bark, think to themselves, oh, I'm underground now and I need to produce roots. So they produce roots, and then you can take that whole branch, cut it off from the main plant, put it in a pot or plant it somewhere else, and you have propagated a second organism from that mother plant. You can do this on multiple stems on the same plant and get many propagated new individuals of that species by air layering. Trench layering is also kind of cool, where you take a branch and basically kind of almost bend it, a long branch, bend it in a spot till it slightly breaks, not completely breaks, but just bends until you kind of see the bark cracking and a little bit. That's wounding it enough to then put that underground. The red little candy cane like looking things are just like a staple or a piece of wire that holds it underground. And after a while, roots will form off of that wounded area of that stem that's buried, and then you can cut in between those sections that are underground and have new plants. You could also layer them into pots if you wanted to. So let's talk about a node. This is something that'll come up a lot, and we'll get into it more when we do the full-length lecture on cuttings. But a node is a place of growth on a stem, period. It's a place of growth. There's little areas along the stem where cells are dividing, and that's where leaves will grow out of, or a new branch, or a flower. Cells are dividing and making new tissue at a node. Then there's areas in between the nodes, called the internode, where cells are not dividing. There's no new cells being added. In this little picture here, the area between the leaves on the stem, there's no new cells being added there. The cells can stretch a little bit, but there's no growth happening there. But the areas where the leaves come out, there's cell division happening there. And those are called nodes, the place of growth or cell division on the stems. And we want to make sure when we're doing cuttings or layering, we're always focusing on the node, because that is the place of highest probability that roots will form on your cutting and in this layering too. So here you would want to make sure that you have got a node or two that is buried and your wound is near that node. And when you're taking cuttings, you want to make sure you've got a few nodes on your cutting, and a couple of them get buried underground when you plant them, and a couple of them are above ground. Roots will ideally grow off of the nodes that are underground or under the soil, and leaves will grow out of the nodes that are above soil in your cutting. Mound layering, another technique of layering, is where you take say a shrub, cut the plant back in maybe late winter, and then in spring it forms a bunch of new shoots. You can then bury those shoots, mound up soil above ground around that stem and around those shoots, and as the season progresses you can add a little bit more, and roots will form off of those branches that have been buried, and then you can cut all of them off and they become individual plants. There's when we buy rootstocks for grafting, they're produced in this way, and bare root shrubs are often produced in this way, where you can get multiple stems coming off of a plant. They can be made into new individual plants. Kind of imagine when you cut something back, often it will shoot out a whole bunch of stems in reaction to being pruned, and then you can take advantage of all those stems that have been shot out and produced by burying them and mounding up around them and getting them to produce roots. Divisions, another asexual propagation method. Really simple. Plants that grow in clumps and get bigger and bigger and bigger. Think of like grass clumps, big ornamental grasses, not turf grasses. Bamboo, bird of paradise. There's just so many plants that grow in clumps, and as they age the clump gets bigger and bigger, and at some point they're so big that the center dies out, or they've absorbed all the nutrients in the soil, they're too big for the spot. You can dig them up, divide them up into smaller subsections, and then replant those, or throw some away or give some away. But that's what a division is. If you're a propagator at a nursery, then some plants are very easily propagated using divisions. Some of the plant forms or organs that plants have that lend themselves to divisions are plants that produce bulbs, corms, which are very much like bulbs, rhizomes, they're underground stems like an iris or ginger, tubers, runners. Think about strawberry plants with runners, these horizontal stems that go out on the ground on the surface of the soil and root. You can divide up those. Stolons, which are similar to runners, and even suckers. So all of these types of plant forms and organs lend themselves to using divisions for asexual propagation. Grafting is a really fun and interesting method of propagating plants asexually for cloning them. We will be doing this in class and learning more about it later. It's joining two parts of two different plants together. So you take the scion, which is a branch or a stem from a portion of a donor plant, and that is grafted or joined to a stalk or root stock. And you can see the little diagram here. The scion and the rootstock are joined up. They're from different plants, but when you put them together and kind of seal up that connection there, they will grow together and form a new plant. And the top of the plant, which comes from the scion, will have all the genetics of that scion wood, and the bottom will have all the genetics of the rootstock. So think about avocado trees. All of our avocados in the south coast here, they are all grafted. The rootstock is an African rootstock that is resistant to root rot, so it can handle heavier soils that aren't super well drained. We have more clay around here. And then the scion wood is the wood that you use in this graft that produces the kind of fruit that we want, Hass or bacon. There's a bunch of other, Fuerte is another avocado type. So to make sure that your tree produces that exact type of fruit, you would graft. You take a branch from an existing tree of a scion wood and graft it onto the rootstock. And then you have a plant that produces the fruit you want and is hardy and adapted to that climate and soil. Tissue culture, the final method of asexual propagation that I'll mention in this overview. It's pretty simple. You just take a little bit of a tissue of a plant, and you can just take a little bit, teeny bit. I propagated ferns by tissue culture when I was at UC Davis, and we would take a hole puncher, like a single hole puncher, and just punch holes, a little round disc of tissue from the leaf of a fern, and then you put it on some kind of gelatin or agar media. So there's like a little petri dish in the top picture, a test tube in the bottom picture. Little bits of tissue from a mother plant are put on top of those that agar, and in that agar are nutrients and hormones and things that will induce that little bit of tissue to grow roots and then shoots and produce a whole new plant. So it's a really popular and great way to get a lot of plants, new plants, in a short period of time. But it does have to be done in a laboratory setting. Everything has to be very sterile. But all of these little clones that are produced from the tissue are true or identical genetically to the parent plant. It's a very common method for producing ornamental landscape plants these days, getting more and more popular. Some of our nurseries here around Santa Barbara will often grow little starts, they'll call them seedlings, of plants from a tissue culture company, have them shipped to them, and then they pop them up and grow them up for a few months till they're a size and maturity that can sell. Tissue culture is also called micropropagation. I mentioned using little discs of leaf tissue, but often the very tips of shoots, the very ends of a branch, the terminal shoots, or small immature leaf buds are cut off the mother plant and put onto this agar gel, and that has nutrients in it and hormones, and you can produce thousands of plants quickly. You can see on these petri dishes here, like there's like about 20 little seedlings growing here, whereas if you were growing them from seed or from cuttings, you'd need like a pot and a lot more space for each of those to grow up. So this is a very efficient, space efficient and time efficient way to propagate and clone plants asexually. Just so you know, genetic engineering, I know you've heard of that. It's when genetic material from one organism is moved and inserted into the genetic material of a different organism to modify that second organism. That's genetic engineering. And why I bring it up now is it uses the methods of tissue culture for this to happen. So if you think back to the pictures on the last slide, you've got all those teeny little plants on a petri dish. Even before they grow roots or shoots, they're just a little mound of cells that's dividing. At that point is when, if you were doing genetic engineering, you would get DNA from your donor organism. Let's say you're trying to have this plant that you're looking at here looks like, pretty sure this is a young tobacco plant, and this was actually done. The genes for phosphorescence or glowing were taken from fireflies, the genetic material. They are extracted, put into the DNA of a bacteria, and a kind of a bacterial soup is formed that has a lot of that specific genetic information, in this case for bioluminescence or glowing. And then that bacterial liquid that contains those is spread on the tissue of the tissue culture, those little mounds of growing cells of the plants. Some of those plant cells, because they're very immature, will absorb the genetic material from the bacteria. Here you see the end product is young tobacco plants that are bioluminescent. They glow because they have that gene from the firefly that was transferred using the methods of tissue culture and a bacterial vector which transferred it over to it. So almost, well, you might already know this, but almost all the bread that's out there on the market, beer, wine, cheese, lots of the fish that we buy, including shellfish, they are all genetically modified organisms. So this has become a very important method, this tissue culture, in a very, very lucrative industry. Advantages to asexual propagation. All these methods we've talked about, cuttings, layering, divisions, tissue culture, grafting, you can produce more mature plants in a shorter period of time than if you used seeds. And a lot of seeds are not always viable and don't always produce the same characteristics as their parent. So that's why asexual means would be advantageous. Some of the downsides of asexual propagation is you need some special equipment and skills, but as you'll find in this class, they're not that complicated, and they're kind of fun because of that. The cuttings involve detaching a piece of tissue from a parent plant and then inducing it to form roots and shoots. So during that time that it has no roots or shoots, it's very vulnerable, and it often will rot or desiccate, dry out, and it doesn't work. So that's the hard part, because it's a very kind of harsh part of the process for the plant to live through. Also, some plants are patented, meaning you cannot propagate them through these means. We will talk more about that. And as we propagate some of our plants, and some of our plants that we'll propagate in this class they actually are patented, but we have authorization from the grower to propagate them, just not to sell them. And some basics in propagation that you will practice and learn, but I'll go over them now. Some of the tools you'll need: seed cleaning sieves if you're gathering wild seeds and cleaning them and storing them and using them for propagation. Dibblers are small little tools that make seed planting easier. There's other seed sowing devices on a larger scale. You'll need knives for grafting and taking cuttings. Clippers are great for taking cuttings. Grafting tape is required too. So there's some special equipment, but you can do a lot with just a set of clippers. Some basics on the media, and we just call it media. It's just the soil type that we put our cuttings into, or seeds, or whatever we're propagating by whatever method. There's different types of soils. There's sandy soils, there are clayey soils, alkaline, acidic, peaty, silty. Depends what you're growing. It's all going to be dependent on what you're growing, which type of propagation media you choose. When you're doing it on a commercial scale, it's best to always use sterilized soil. You can purchase it sterile because you don't want to have pathogens in it. If you're doing it for yourself, you can use your garden soil, and some of your cuttings and things might die, but it's not a big deal if you're just doing it for yourself. But if you're working for someone else and they said, I want 500 Mexican sages to plant in November, and it's September, you want to use a method in the soil that's going to ensure that you are able to propagate that many by that time, and part of that would be using sterile soil, so you don't lose a lot of them to some kind of soil pathogen. There's a list of different kind of amendment soils. and propagation media can have in them for different reasons. And if you're growing native plants, you're going to use certain recipes for that growing media as well. So it just depends on what you're using. Propagation environment is important too. The kind of environment you provide for your cuttings or your divisions or your seeds. You've got to provide the right moisture level through some kind of irrigation system, a hand system or an automatic system. Temperature needs to be controlled through heating mats, warmed benches, a heater in your greenhouse or house, or a cold frame. The soil needs to be well aerated for many types of species. The pH of the soil and water should be monitored. You don't want it to get too high or too low. Nutrient levels need to be kept at an adequate amount in the soil once the plants have roots and leaves. Humidity should be fairly high inside enclosed areas like greenhouses or hoop houses or cold frames, but it needs to be humid air, but moving humid air. You don't want still humid air just sitting on the plant. That will cause rotting and pest outbreaks. But humid air and ventilation, fans, maybe an open structure, things that keep that air moving around will deter pest growth. And proper lighting. Maybe your plants might require just full sunlight or partial shade, and they should be given that when you are propagating them. And sometimes plants require extra light. I know one year we grew dahlias in this class, and so we installed lights in the greenhouse and kept them on for 16 hours a day is what was needed to get them to flower pretty quickly. So